So today I'd like to talk about safety aspects as relating to microscopy, and specifically I'd like to talk about safety aspects when collecting and also growing microorganisms that you would like to put under your microscope. So hello and welcome again, Microbehunter here. My name is Oliver. And today I wanted to talk about some safety aspects concerning microscopy. Of course, I do not want to talk about the danger of cutting yourself with a sharp knife when you prepare specimens. I mean, this is a very well-known danger, of course. And I also do not want to talk about the possibility or the dangers of inhaling, let's say, certain hazardous substances when you're preparing microscope slides. I mean, there are plenty of solvents or stains even that are used in microscopy. No, these are not the topics, even though I think they're also important. But rather, I'd like to talk about the issue about growing microorganisms or simply collecting microorganisms for microscopy. And I would like to start off with a little story here. It's not a story, it's actually something that really happened. Because many years ago I was doing microbiological work at a university. I was working for my master's thesis, and my task was the identification of bacteria. So we collected bacteria. And my job was now to do a whole range of biochemical tests on those bacteria to determine what they are. And so I had to grow them. I had to grow them in flasks, in liquid culture medium. I was also growing them on agar plates. And hundreds of them, really. Those bacteria, of course, I did not know what they were. That was the whole point of my thesis, is to figure out what are they, and I was therefore growing unknown bacteria in liquid medium and also on petri dishes. And we also had an inspection, a safety inspection once. So the guy came into our lab and he looked at all of the petri dishes standing there on our tables, and he was really, really unhappy about this. And he said, "Come on, guys, you're working in an elevated biohazard category, biohazard category 2. There are 4 categories, and the 4 is a very dangerous one where you have to work in space suits almost, yeah. And category 1 is totally, it's not dangerous." But because we were working with unknown bacteria, this was an elevated safety category of biohazard level 2. And we were just working there in regular lab coats and so on. So and of course we were using gloves and so on. But in order to grow unknown microorganisms, unknown bacteria, you had to be an authorized lab. You cannot simply do this in any lab, but you had to be an authorized lab. And of course, there were also those inspections. And you see that issues like this, these are taken seriously. And so the guy kind of advised us on all safety issues that we had to take care of. And now what happens now when, for example, when I turn on the computer and watch some YouTube videos, occasionally I do see people who are growing their own microorganisms, their own bacteria on petri dishes at home. And what they sometimes are doing is it's a nice demonstration, really. They're making their own agar plates using a growth medium, or you can even buy them over Amazon. And then people are making fingerprints. And those fingerprints you can then see if the bacteria on your finger, then those bacteria transferred on the agar plate. So you're kind of pressing your thumb against the nutrient medium in the agar plate. And after a couple of days, you're going to see that there are bacterial colonies growing on the plate. And each colony contains billions of bacteria. And this kind of shows how many bacteria there are on your skin. It's a nice demonstration activity that sometimes people do. But every time when I see this on YouTube, I'm not really happy, because you're doing this at home. Actually, you have to be an authorized lab to do that. And the second thing that I'm a little bit more concerned about is, you are actually growing bacteria that have the potential to grow on your skin. And these are potentially pathogenic. That means disease-causing bacteria. And not only that, but you're also growing them to extremely, extremely high concentrations, like concentrations which you normally naturally would probably rarely find in the environment. And this is, of course, a combination of factors that I would consider already problematic. First of all, the high concentration, and number two, the type of bacteria which have the possibility to grow on the human body. And for this reason, I would not advise people to actually grow their own bacteria, especially if they're isolated from your own skin, at home. Now, how is the situation now if, for example, you want to observe some microorganisms that you've, let's say, isolated from a home aquarium? I mean, there's many people have a home aquarium, and there are some algae maybe growing on the glass, or maybe on the of the aquarium, or maybe in the sediment. And wouldn't this be interesting to put under the microscope? And I would say, of course, by all means do that. Yeah. But you have to understand that the microorganisms that grow there, you have not isolated from human skin. And also the concentration is much lower than, for example, when you grow them on an agar plate. So I would say that I'm not saying it's completely free of problems. I still would not eat those things, and I would also take care that I do not have any wound or any cut in the skin before touching it. But generally, I would say that this is not as big of a problem as growing microorganisms, bacteria, which have the possibility to grow and inhabit the human body. Okay, so this is one thing. And number two is when you isolate, for example, microorganisms from a pond or a river where people are also swimming. And if it's clean enough to swim and maybe you scratch some of the material off from the surface, then I would also say that this is, of course, less of a problem than, for example, if you are to put polluted water or sewage water under the microscope, where it's clear that there are many bacteria in there that actually come from the human body, from the human digestive system. Yeah, after all, it's sewage water. Yeah. So what I'm trying to say here is that both concentration and type of bacteria do play an important role here, and you should take this, of course, into consideration. Why am I saying this? Because occasionally I get emails from people who've collected some water samples, let's say from a local pond or river, and it's a reasonably clean pond or river, and then they're kind of worried that they have now microorganisms in the jar, and then they ask me, okay, is this dangerous now? And I'm saying, well, if it's in a jar and if it's not decomposing or breaking down or anything, it's probably not more or less dangerous than, let's say, taking a bath in that pond, right? Because when you're taking a bath, you're also exposed to the very microorganisms. There are, however, some ponds that are so polluted or that are extremely rich in so-called cyanobacteria. And cyanobacteria are what used to be known as the so-called the blue-green algae. And some of them actually do produce quite potent toxins. So you wouldn't take a bath in those ponds anyway. Yeah. Or for example, if there are lots of birds in a pond and if the birds pollute the water. There's salmonella possibly there. Salmonella bacteria. It's also not something that I would like to get in contact with. So what I'm trying to say here is a little bit of common sense, of course, also plays a role here. If the water is sufficiently clean to have a swim, then I would, of course, be less concerned than if the water, of course, is polluted. So this is also something that I also then transfer then to my microscopy work at home. If I have a jar with relatively fresh and clean pond water and many water fleas and algae which eat bacteria and paramecia that also remove bacteria, and the bacterial concentration is low, then of course it's a different thing than if it's a smelly water from quite a dirty pond, obviously. So what I'm trying to say here is that you cannot generalize this. But as I mentioned already before, the type of microorganisms that are there and also the concentration both play a very important role. Now, occasionally you can read online or hear videos where people suggest the making of a so-called a hay infusion. This is, I think in the 19th century, people used to do this quite a lot. You take some dried grass and you put it into a jar with a little bit of water. And then you wait for a few days. And the first thing that's going to happen is that the water changes its color, turns brownish greenish, and then it starts to turn turbid and cloudy. And this is a sign of a lot of bacterial and microbial growth. And people in the past, in the 19th century, they put this under the microscope and they were able to see a lot of different microorganisms there. And this is not something that I recommend. Because indeed the bacterial concentration is very high here and you do not know what you're really growing. Now I know that I just said, well, those bacteria are not from the human skin and so on. But still, I think it is not necessary to do that, and because the amount is quite high that you have. And some people say now, okay, I want to dispose of this, and then they're dumping it in the toilet, and then you have the formation of aerosols. They're little small droplets. So all of the bacteria actually spread through the air. And what you can do, or what some people think that they can do, is they say, okay, I'm going to drop a little bit of alcohol, disinfecting it into the liquid. But if you do that, you should know that, of course, the alcohol becomes diluted very quickly. So you have to use actually a fairly large amount of alcohol. Or you use some kind of a chlorine disinfectant, which is, by the way, is also not the very best for the environment because of chlorine. I just wanted to make it clear here that this is also, disposing of this is then also, I wouldn't say a big problem, but something that you might take into consideration, that if you just pour it into the toilet or somewhere, I mean, you've got a little, droplets forming that with bacteria that you're going to inhale. So it is not something that I generally would recommend doing. And so these hay infusions, I personally also think are not always the most interesting ones, because they contain a lot of bacteria. Actually paramecia you won't find in there, because paramecia are not able to survive dryness. They do not form cysts that are able to survive dryness. So I just want to discourage people of actually using hay infusions. Now, if you want to use safe microorganisms for observation, then I highly recommend that you use those bacteria and fungi which you can eat. Yes, and I'm not talking, obviously it's not spoiling of food. You cannot eat those. It's very dangerous, as a matter of fact, because those microorganisms can form toxins and can make you ill if you eat, for example, moldy food. But you can, for example, buy yourself some cheese that contains mold on the surface. And this is mold that has been added by the manufacturer of the cheese, and is something that you can put under the microscope, especially when you want to do microscopy with children. Then this is something that I recommend. Or if you want to observe bacteria instead of spoiling food, I highly recommend that you use yogurt bacteria. And you can even buy freeze-dried yogurt bacterial cultures, let's say over Amazon or so. This is then safe for observing. After all, safe enough to eat, right? So fun. Or, for example, yeast is another example of a fungus that you can. Baker's yeast is used for cooking. So I'm trying to make it clear here that there are many safe sources also available, which does not make it necessary to grow microorganisms at home. Now, what I have done already, of course, also if you watch my YouTube videos, I've been enriching paramecia quite frequently. So I take a water sample with some algae in it, and I add a crushed wheat grain. When you do that, then after about 3 or 4 days, a lot of paramecia can be found there, because the wheat grains serve as a food source for bacteria, and then the paramecia will eat the bacteria, and then you also have a lot of paramecia there. Now, when you do things like this, then make sure that the oxygen concentration stays high. And the reason why this is so important is because otherwise bacteria are going to increase too rapidly. Paramecium and other ciliates require a lot of oxygen, and if the oxygen concentration drops, and if the bacterial concentration increases too quickly, then the paramecia are going to die off, and they're also not able to keep the bacterial concentration low. And for this reason, I encourage you that you do not put the jar into the bright sunlight where it's going to heat up and then this drives out the oxygen again, but keep it rather in a bright place so that any algae that can be found in there actually do photosynthesis and produce oxygen. And then also the paramecia have an easier time growing, which keep the bacterial count low. Because that's actually what you want. You want to keep the bacterial count as low as possible. And as soon as a jar with water sample starts smelling, maybe it smells a little bit foul or it smells a little bit like sulfur, then you know that the sample has become anaerobic and decomposition started because of the bacteria. And that's something that you should of course avoid, because every time when you have bacteria increasing in number, you really don't know what you're growing so much, right? Are they dangerous or not? Yeah, you don't know. I remember that when we had this inspection that I talked about a couple of minutes ago, then I asked the guy, and he says, well, why do you assume that they're dangerous? You know, I mean, we isolated them from a natural environment and not from the human body. So why do you assume that they're dangerous? Right? And then he responded like this: why do you assume that they're not dangerous? You know, you don't know what you're growing, really. And he's actually correct, because we were growing those bacteria also in flasks of liquid nutrient medium. Yeah. And in the lab, things can happen where you drop a flask and then you have microscopic droplets of bacteria flying around the lab, and we inhale them and we actually don't know what they are. And that is the issue that we have to be simply aware of here. Yeah. So in summary, use your common sense a little bit. Have a healthy. How do you call this? Be cautious. A healthy amount of cautiousness, but don't be overly paranoid. But try to keep the concentration low, and do not deliberately grow bacteria from your skin, or do also not decompose, let's say, food material like meat or so. It's not a good idea. Also avoid sewage water. Some people, want to observe this as well. Come on. Microscopy is a hobby. And what we want to use safe sources, and there are plenty of safe sources around in any case. Yeah, but I'm going to leave it at that. I hope it was interesting for you. Leave comments behind, please. Bye-bye. See you around next time, and happy microbe hunting as always. Bye-bye.